Hello my dear friends, welcome to Vidya Aradhana Academy, a premier institute for NEET and JEE coaching which is located in Latur, Maharashtra. Myself Dr.Y.N.GajapatiVarma. Today in this session we are discussing the previous year questions from the very important chapter, and the chapter that is having very high weightage in NEET examination, that is nothing but principles of inheritance and variation. So here we are discussing the previous year questions from 2016 to 2020. So now let us quickly go through these questions. See here, the first question is, how many true breeding pea plant varieties did Mendel select as pairs which were similar except in one character with contrasting traits? So here what they are saying is, so Mendel in total worked with how many total varieties of pea plant? So here very important, Mendel actually worked with 7 characters, very important, and each character has 2 traits. One is dominant trait, another one is recessive trait. So 7 characters and each one is having 2. So 7 into 2, total 14 different varieties or 14 different traits. And please, very important point, Mendel obtained a pure breeding plant for each trait. So that's why he got actually 14 different pea plant varieties. So the answer for this is option 3, that is 14. Next one, identify the wrong statement. We need to identify the wrong statement here with reference to the gene I that controls ABO blood groups. We know that the gene that is controlling the blood group is indicated with I. I actually refers to isoagglutinogen. Okay, so here the first statement is, the gene I has 3 alleles. Yes, it is true. IA, IB and small I we are representing. Sometimes the small I can be represented as IO also. Next one, a person will have only 2 of the 3 alleles. This is very important point. Very important point, because in the diploid individual, for a particular chromosome, 2 will be there. One is obtained from the father, that is called paternal, and the other one is obtained from the mother, and that is called as maternal chromosome. So in this way only 2 chromosomes are present. So that's why any gene can have a maximum of 2 alleles only. So here this is another important concept that is called multiple allelism. Multiple allelism can be observed in the population. More than 2 alleles we can observe only in the population. In one individual, maximum we can see only 2 alleles. So this is also correct statement. When IA and IB are present together, they express the same type of sugar. This is actually a wrong statement, because when IA is present, that is producing an enzyme. That enzyme will add N-acetyl glucose amine to the existing O antigen or H antigen or H substance. So here it is adding N-acetyl glucosamine. Whereas when IB gene is present, it is producing an enzyme. That enzyme will transfer simply galactose. That's it. Here N-acetyl galactosamine, but here it is transferring only the galactose. So in this way these two are producing different ones. So that's why in the presence of IA gene it is producing A antigen, in presence of IB gene it is producing B antigen. So these two are completely different, but here they are saying that they will add same type of sugar, and that is wrong statement. Next one, allele small I does not produce any sugar. Yes, it is correct, because when the gene is recessive small I, it is not coding for the enzyme. So since it is not coding for the enzyme, no sugar molecule is added here. So that's why this statement is also correct. So what is the wrong statement here? It is option 3. Clear? Next one, experimental verification of chromosomal theory of inheritance was done by, it is done by Morgan. Very important. Experimental proof with the help of Drosophila melanogaster was done by Morgan. T.H. Morgan. He is also known as father of experimental genetics. Okay. Next one. Select the correct match. Which one out of these 4 was correctly matched? That we need to identify. Phenylketonuria, autosomal dominant trait. It is a wrong statement, because phenylketonuria is a disorder that will be observed during this autosomal recessive trait. Okay, under homozygous recessive condition we will observe this. So it is present on autosomes. So it is autosomal recessive trait actually, but they gave dominant, so it is wrong one. Next one, sickle cell anemia, autosomal recessive trait and chromosome number 11, and it is correct. Yes or no? The beta chain gene is present on chromosome number 11, and here under the recessive conditions, homozygous recessive condition will cause this sickle cell anemia condition. Yes or no? And it is also lethal. So that's why sickle cell anemia is also example for recessive lethal condition. Yes, anyhow, those are some other points that you need to remember. Next one, thalassemia. Thalassemia is due to autosomal recessive, but they gave X-linked. And here hemophilia is X-linked recessive, but here they gave Y-linked. So here 1, 3 and 4 are incorrectly matched, whereas the option 2 is correctly matched. So answer for this is option 2. Okay, so these are some important points so that we can easily remember all these things. There are many chances for asking questions on these simple concepts. Next one, in Antirrhinum, that is snapdragon, a red flower was crossed with a white flowered plant, and in F1 generation all pink flowers were obtained. This is regular cross only, whatever they are giving. When pink flowers were selfed, the F2 generation showed white, red and pink flowers. This is also the regular crossing and the same data they have given. But here what they are asking is very important. Choose the incorrect statement from the following. So what is incorrect actually? That we will see here. Law of segregation does not apply in this experiment. Very, very important. Law of segregation is a universally applicable law. Very important. Mendel proposed 3 laws. Out of these 3, which of these is universally applicable? Suppose they are asking like this. It is law of segregation. Everywhere it is applicable. And the only rare event that is called non-disjunction is there. So that is only exception, but that is a different case. Okay. So please remember. So it is applicable everywhere, but here they are saying it is not applicable. So this is the wrong statement, but anyhow we will go through remaining statements also. This experiment does not follow the principle of dominance. Yes, here in heterozygous condition one is dominant, another one is recessive. That concept is not applicable. So it is an example for incomplete dominance, no? So that's why this statement is correct. Next one, pink flower in F1 is due to incomplete dominance. Yes, it is correct. Next one, ratio of F2 is 1/4 red, 2/4 pink and 1/4 white, and that is also true. So here the incorrect one is the option 1. Okay, very important. Next one, what map unit or centimorgan is adopted in construction of genetic maps? Very, very important concept it is. See, in these genetic maps construction, whenever we are using distances we are indicating in simply units or map units or centimorgan. If we clearly see this centimorgan, so here in this centimorgan, what is the basic 1 centimorgan means what? Here what they are saying is, what is the definition of that unit or map unit or centimorgan? 1 centimorgan means 1% crossing over. So if between 2 genes 1% crossing over is occurring, that is equal to 1 centimorgan. Suppose 10% crossing over is occurring, 10 centimorgan. Yes or no? 30% crossing over is occurring, 30 centimorgan or 30 map units. The same thing they are asking. So the unit of distance between the genes on chromosomes representing 1% crossing over. 1% crossing over means 1 centimorgan or 1 map unit. This is what the definition is. Okay, so simply the definition they have asked here. So answer for this will be definitely option 4 only, because in other they are saying that 1 centimorgan is 50% crossing over, 10%, 100%. So that is wrong. Okay, so please remember this one. Next one, the frequency of recombination between genes present on the same chromosome as a measure of distance between the genes was explained by. So here what they are doing is, what they are saying is, actually who developed these genetic maps? How we are preparing the genetic maps? Yes, as the distance increases, the percentage recombination increases, percentage recombinants or crossovers will be more. So in this way, if the percentage recombination is more, distance between the genes is more. So in this way, one scientist whose name is Alfred Sturtevant, actually he used this concept to prepare the genetic maps. So that is what the question they are asking. So who developed this one? It is option 4, Alfred Sturtevant. Very, very important. Next one, select the incorrect statement. So wrong statement we need to identify here. Male fruit fly is heterogametic. Yes, it is correct. Please remember, fruit fly is nothing but Drosophila. So already for the incomplete dominance in the NCERT they have given one image, no? So there they have clearly mentioned female is having 2 X chromosomes, male is having 1 X and 1 Y chromosome. Y chromosome is having hook also. Yes or no? Clearly mentioned in the NCERT in the form of diagram. So here male fruit fly is heterogametic. This statement is correct. One X and one Y are present, so it is heterogametic. It will produce different types of gametes, two different types of gametes. Yes or no? So it is correct. Male grasshoppers, in male grasshoppers, 50% sperms have no sex chromosome. This is also true because in the case of grasshoppers, males are having only one sex chromosome, females are having two sex chromosomes. Yes or no? So here if we see whatever is the autosomal set is there, so then XO is present for the male. In the case of female, two autosomal sets are present. Autosomes are present as pairs, and here female is having two sex chromosomes. So here when the male is producing the gametes, now here X will go here and one autosomal set will go here, and here one autosomal set and there is no sex chromosome. Here 0 means there is no sex chromosome. That means two different types of gametes are produced. But in the case of a female, A plus X and A plus X. So females are homogametic and here males are heterogametic. And very important point is, in the case of grasshopper males, here number of autosomes is same in both of them. Number of autosomes is same in both of them. With respect to sperms we are talking. But 50% sperms are having one sex chromosome, but remaining 50% do not have any sex chromosome. Okay, they are not having any sex chromosome. So in this way, this is the point that they are actually saying there. Okay, so here, so this is what they are saying here. In male grasshoppers, 50% sperms have no sex chromosome. So this is also correct statement. Next one, in domesticated fowls, sex of the progeny depends on the type of sperm rather than the egg. So here what they are saying, it is dependent upon the sperm. See, when it will be dependent upon the sperm, if the male is heterogametic, then the sex of the next generation is dependent upon the male. Suppose the female is heterogametic, sex of the next generation is dependent upon the ovum or the egg. Yes or no? So here they are talking about the fowls, means birds. So in the birds, who is heterogametic actually? Yes, it is the female which is heterogametic. When the female is heterogametic, then the sex of the next generation is dependent upon the ovum, because a female is producing two different types of gametes, but the male is homogametic. So male will not decide the sex of the next generation. Female will decide the sex of next generation. Yes or no? So that's why this statement will be wrong. Next one, human males have one of their sex chromosome much shorter than the other. Yes, this is also true, because in the humans, two sex chromosomes are present. One is X and another one is Y. Y chromosome is much shorter when compared with the X chromosome. So this statement is also correct. So what is the wrong statement here? So it is the option 3. Yes or no? So in this way, with this question is completed. Next one. What is the genetic disorder in which an individual has an overall masculine development, gynecomastia, and he is sterile? Yes or no? Masculine development, so he is developing into the male. Male and sterile, then gynecomastia is present. So what is that individual? It is Klinefelter syndrome. Please remember here, very important. In the case of Turner syndrome, these are very important. These are very important. In the case of Turner syndrome, what happens is number of autosomes will be there, and here XO is present. Please remember the very important point. In the case of humans, how the male and female are determined, sex determination. If Y chromosome is present, that's it. It will develop into the male. Here, for the development of female, absence of Y chromosome will determine. Please remember here, don't think that two X chromosomes are required. Even though single X is there, yes, it will definitely develop into the female. Please remember this concept. Here, absence of Y chromosome will automatically or by default the fetus will develop into the female. Please remember, this is very important. So that is the reason why Turner syndrome, always females. Turner syndrome is seen in always females. Whereas Klinefelter syndrome. So in the case of Klinefelter syndrome, please remember here, even though two X chromosomes are present, presence of Y chromosome will make this into male. So that is the reason why simply presence or absence of Y chromosome will determine whether a particular fetus will develop into male or female. This is very important point. So that's why Turner syndrome always females, Klinefelter syndrome always males. Yes or no? So this is another important point that we need to remember. And here they are also mentioning about the Edwards syndrome. Edwards syndrome is a trisomy of 18th chromosome and Down syndrome is a trisomy of 21st. See, these are the trisomies of autosomes. So these can be seen in both males and females, whereas these are fixed. Yes or no? Please remember this point also. So for this question, what is the answer? Klinefelter syndrome. Next one. Which of the following muscular disorder is inherited? So here actually in the NCERT under the pedigree analysis, for the first pedigree, at the legend of the pedigree they have clearly mentioned. So it is autosomal dominant. Within brackets they have given myotonic dystrophy or muscular dystrophy they have given. So this is genetically inherited, whereas the remaining are not related to the genetics. Okay, so that's why the answer is option 2. Next one. Which of the following characteristics represent inheritance of blood groups in humans? So here they have given 5: dominance, codominance, multiple allelism, and incomplete dominance, and polygenic inheritance. Here if we clearly see, out of these, how many are actually related to human blood groups? Very simple concept, but very important. Here actually we are studying about 3 different alleles. Here we are studying about 3 different alleles, IO, and this can also be written as small I. If we clearly observe here, IA and IB alleles, these are showing codominance. They are showing codominance. Okay, and each of this is again completely dominant over the recessive allele. That means IA is dominant over small I. IB is dominant over small I. See, actually we should not write like this. For easiness we are using like this. See, these two are showing codominance, so we are observing codominance, and we are observing complete dominance, or simply we can call this as dominance. Dominance or complete dominance, or sometimes this can also be called as dominance recessiveness. Yes, one is dominant and the one is recessive. That's why we can call this as dominance recessiveness also. Okay, so in this way these we are observing. And anyhow, 1, 2, 3. Since more than 2 alleles are present, this is an example for multiple allelism. So these are the 3 things that we are actually observing here. So 1, 2, and 3 are correct. Incomplete dominance we are not observing. This is not due to polygenic or quantitative inheritance. So that's why 1, 2, 3 is correct. So that's why option 2 is the correct one. Okay, next one. A woman has an X-linked condition on one of her X chromosomes. This chromosome can be inherited by. So it will go to whom? So will it go to only the grandchildren, and or it is going to only sons, only daughters, or both sons and daughters? That's what they are asking. See here, it is an X-linked condition. So X chromosome of female is going to whom? So this is what actually we have to observe here. Yes or no? So in the case of a female, 2 X chromosomes are present. Suppose here she is married to a man. Okay, now if we clearly see this one, here what happens? Here X chromosome, like this we will write. Okay, so here X and X, X and X, now X and Y, X and Y, now again X and X, and then X and Y. Okay, so here we are talking about each and every X chromosome. Okay, so here if we clearly see, this is a female, and this is male, female, and male. So if we clearly see, the X chromosome of female is going to next generation daughter. So female in the next generation is daughter. Okay, and not only that, the X chromosome of female is going to next generation male. That means it is going to the son. Yes or no? So similarly for this one also it is going. So that means X chromosome of female or mother can go to next generation daughters or sons. So both of them, there are chances for both the possibilities. So anyhow, that option also it was given here. So it is the 4th option. So both sons and daughters can get X chromosome from the mother. But please remember, X chromosome of father will go only to the daughters. Sons will never receive X chromosome from the father. They will receive only from the mother. So this is another important point. So another question is also there based on this. So that also we will go through here. Which of the pairs is wrongly matched? Incorrect matching we need to identify. XO type of sex determination, grasshopper. Yes, this is correct. Next one, ABO blood grouping codominance. So here just now we studied no codominance is there, dominance is there, both of them are there. So anyhow codominance they have given, this is also correct. Next one, starch synthesis in P. It is due to multiple allelism that they are giving, but this is the wrong one. Okay, anyhow that I will give you the explanation. T.H. Morgan linkage. Yes, he actually discovered the linkage. So now we will see regarding that starch synthesis and multiple alleles. Please remember here regarding the starch synthesis in the NCERT they have used this capital B and small b. Yes or no? So here we are studying about only two alleles, capital B, capital B, capital B, small b, small b, small b. Due to more production of starch branching enzyme, large quantity of starch is present and the seed size is large. Okay, so in the homozygous recessive condition, small b, here what happens? So here very less quantity of starch branching enzyme is produced and the seed size is very less, so that it is having small sized seeds. But here please remember, with respect to seed size it is showing incomplete dominance. Incomplete dominance. So because of this here it is giving intermediate size. Intermediate size. So in this way only with respect to two alleles only we have studied. We did not study any multiple allelism here. So that is the reason why this is wrongly matched. So the answer for this is option 3. Clear? Next one. Among the following characters, which one was not considered by Mendel in his experiments on P? So this we can easily see. See, regarding the stem height, tall and dwarf, he studied. Regarding the pod shape, inflated or constricted, he has studied. And regarding the seed, so whether it is green or yellow colored, that also he studied. But he did not study regarding the trichomes, whether they are glandular or non-glandular. So that's why the answer is option 1. Okay. Next one. Which one from these given below is the period from Mendel's hybridization experiments? In the NCERT, this duration was clearly mentioned. It is from 1856 to 1863. So direct NCERT point. So that is the reason why you have to remember years and the names of the scientists also, at least those which were mentioned in NCERT. Okay. Next one. The genotypes of husband and wife are IA, IB and IA small I. Among the blood types of their children, how many different genotypes and phenotypes are possible? For this we need to do the simple genetic cross so that we will easily get the answer. Okay, first remember those genotypes. Now we will try to do the genetic cross here. So one is having the genotype that is IA, IB. Okay, and the another one is having the genotype IA small I. Is it clear? So now if we are preparing the genetic cross, now here two types of gametes are produced, IA and IB, and here also two types, IA and small I. So when we are doing these crosses, so simply you just do the cross like this, and now we will write the genotypes here, IA, IA, and this one will be IA small I, and here it will be IA, IB, and here it will be IB small I. So in this way, if we clearly see, 4 different genotypes were obtained. 4 different genotypes were obtained. IA is A blood group, and at the same time IA small I is also A blood group. So this is A blood group, and this is AB blood group, and this is B blood group. So here if we clearly see, 4 different genotypes and 3 different phenotypes were obtained. So where is that option? So that we will identify here. So here 4 genotypes, 3 phenotypes. This is the correct option. So that's why the answer for this question is option 2. That's it. Next one. Thalassemia and sickle cell anemia are caused due to a problem in globin molecule synthesis. Select the correct statement. So this is very important. In the NCERT, at the end of this thalassemia, they have mentioned clearly regarding the relation between thalassemia and sickle cell anemia. Thalassemia is due to decreased production of these globin chains. Yes or no? So this is due to decreased production. So that's why thalassemia is quantitative problem. So clearly they have mentioned. Whereas in the case of sickle cell anemia, there is a point mutation. Because of this, even though globin chain is produced, it is not like the normal one. So it is a mutated one. So that's why sickle cell anemia is qualitative problem. So please remember here, thalassemia is quantitative problem, sickle cell anemia is qualitative problem. So based on that statement they have given here. Both are due to qualitative defect in globin chain synthesis. Both are qualitative, they are saying. It is wrong. Next one. Both are due to a quantitative defect in globin chain synthesis. Both are quantitative, they are saying. This is also wrong. Next one. Thalassemia is due to less synthesis of globin chains. That means it is quantitative problem. Yes or no? Less synthesis. Yes. So this statement is correct. Next one. Sickle cell anemia is due to a quantitative problem of globin molecules. Sickle cell anemia is not quantitative. It is qualitative. So out of all these three, all these four. So what is the correct option? It is option 3. Understood or not? Next one. A tall true breeding garden pea plant is crossed with a dwarf true breeding garden pea plant. Okay, tall true breeding and the dwarf true breeding. These two are crossed. When the F1 plants were selfed, the resulting genotypes were in the ratio of. It is very, very simple, and the general, whatever we are studying in the NCERT, the same thing they have given here. But the important thing is, in the first option, if we see, so in the F2 generation they are asking, this is the simple general cross only. In the F2 generation, whatever they gave 3:1, tall is to dwarf is correct. But please remember, they did not ask phenotypic ratio. Clearly they asked genotypic ratio. Genotypic ratio is 1:2:1. What is the first one? Capital T, tall homozygous. What is the second one? 2. It is also tall, but it is capital T, small t, heterozygous. So tall heterozygous. And what is this last one? So this one is dwarf, and anyhow it is homozygous. Dwarf homozygous. So that's why please remember here, though option 1 is correct on its own, but for the asked question, option 3 is the best answer, because this is the one that is actually giving the genotypes. Okay, they did not ask the phenotype. So answer for this is option 3. Clear? Next one. In a test cross involving F1 dihybrid flies, more parental type offspring were produced than the recombinant type offspring. Please remember here. See, whenever they are saying that more parental types are produced and less recombinant types are produced, that means there is the phenomena called linkage. Suppose parental and recombinant are produced in the exactly the same ratio in a test cross. Please remember, in the test cross we are not talking for F2 generation. In the F2 generation, definitely more parental and less recombinant will be produced. So here we are talking about the test cross. In the test cross, it is having the equal proportion for all these. So here what they are saying is more parental, less recombinant are produced. That means there is a linkage. So anyhow, in all of this they are mentioning about the linkage only, but the important point, linkage we are observing on. See, please remember, this is also another important concept. Linkage concept is applicable for the genes present on same chromosome. Suppose we are talking about two different genes present on two different chromosomes. Linkage cannot be observed here. The genes present on same chromosome only will show the linkage. Anyhow, these concepts already you know. So that's why based on that point only we need to identify the correct statement. The two genes are linked and present on the same chromosome. Yes, this is correct. Two genes are linked. That is the reason why more parental types are produced. Please remember, here linked does not mean about complete linkage. It is incomplete linkage, because at least some recombinants are formed. So linked means here in this context it is incomplete linkage. Next one. Both of the characters are controlled by more than one gene. So here no. So here in the case of linkage we are talking about one gene controlling one particular character. That's it. Okay. So in this way only we are studying. Next one. The two genes are located on two different chromosomes. No. If they are present like that, they will not show any linkage. Okay. So if they are present on two different chromosomes, since there is no linkage, recombinant and parental will be with equal proportion. Okay. Next one. Chromosomes fail to separate during meiosis. So this is also not correct. Okay. So that's why. So the concept is related to the linkage, and the option 1 is the correct one. Okay. So here, see this one. The genes present on the same chromosome, suppose they are very closely present. Yes, they will show the linkage. Yes or no? So next one. Which of the following most appropriately describes hemophilia? Regarding the hemophilia, very important question. Recessive gene disorder. Yes, hemophilia is recessive gene disorder. That is correct. But go to the next option. X-linked recessive gene disorder. Here they are specifically mentioning that it is X-linked recessive. See here, recessive gene disorder is also correct, but X-linked recessive gene disorder is more correct. Yes or no? Because here they are specifically mentioning whether it is autosomal recessive or X-linked recessive. So in this way, sometimes more than one correct options may be present. In that case, if we want to choose only one option, so then you need to go with the best correct option. Please remember, in the NTA, when they are releasing this notice, they are clearly mentioning that best and correct. So when we are getting some confusion like this, then which is the best one? So which one we will select? So in this way, the best will come into picture here. So X-linked recessive gene disorder is the correct one. Okay. So chromosomal disorder, no. Dominant gene disorder, this is also not correct. So the answer for this is option 2. Okay. Next one. Match the following. So here they have given the matches. So here in the column 1 they have given some phenomena, and the column 2 they have given the explanation. So dominance. So dominance in a heterozygous organism, only one allele expresses itself. That is the dominance. So codominance in a heterozygous organism, both alleles express themselves fully. So that is codominance. Pleiotropy. Pleiotropy means a single gene influencing many characters. Yes or no? Next one, polygenic inheritance means many genes will control single character. So many genes govern a single character. So that's why the correct matching is this one. Okay. Next one. Next one is regarding pick the correct statement. Very important. Correct statement we need to identify. Hemophilia is sex-linked recessive disease. Yes, this is the correct statement. Next one. Down's syndrome is due to aneuploidy. So here in the mutations we have already studied about euploidy and aneuploidy. So what is this aneuploidy? Suppose only one chromosome less or one chromosome more. Two chromosomes less or two chromosomes more. Here entire ploidy is not affected. Only few chromosomes will be affected. Only few chromosomes number may be increased or decreased. If this is like this, that is called aneuploidy. In the case of Down syndrome, it is a trisomy of 21st chromosome. In the normal diploid individual, for every number 2 chromosomes should be there. So in this way, for 21st chromosome, 2 chromosomes should be there. But in the case of Down syndrome, 3 chromosomes will be present. That means 1 chromosome extra is present. So it is an example for aneuploidy. Yes or no? Next one. So that is also correct statement. Next one, phenylketonuria is autosomal recessive gene disorder. That is also correct. Next one, sickle cell anemia is an X-linked recessive gene disorder. This is wrong. Sickle cell anemia is not X-linked, and it is actually autosomal recessive. So out of all these, for A, B and C are correct. So the correct answer is option 4, that is A, B, C are correct. Okay. Next one. So next one is regarding the very important concept of mutation. So here it is about the translocation. Very important point. Sometimes some statements are there, and we are comparing between crossing over and a translocation type of mutation. Please remember, in the case of crossing over, exchange of segments is taking place between homologous chromosomes only. Yes or no? So here in the case of crossing over, exchange of segments is taking place between non-sister chromatids of homologous chromosomes. Suppose exchange is taking place between 2 unrelated chromosomes. So this in this example it is chromosome 4 and chromosome number 20. These two are not homologous. So in this way, exchange of segments is taking place between non-homologous chromosomes. So now there is a complete change in the linkage group. So that's why this is an example for translocation. Understood or not? So in this way, the mechanism that causes a gene to move from one linkage group to another. So what is this linkage group means? Here instead of using the chromosome, they are using another term that is called linkage group. What is linkage group? Suppose you are taking the chromosome number 4. All the genes that are present in this chromosome will form one linkage group. Yes or no? Suppose you are taking another homologous chromosome that is also chromosome number 4. Please remember, that is also having the same linkage group. Both the homologous chromosomes will have same linkage group. So that's why crossing over is exchange of segments within the same linkage group. But here this is chromosome number 4 having one linkage group and chromosome number 20 having another linkage group. So here exchange of segments is taking place between 2 different linkage groups or 2 different non-homologous chromosomes. So this is actually a mutation. This is not crossing over. So here this is a type of mutation called translocation. So please remember this one. Okay. So answer for this is Option 3, translocation. Next one. Which one of the following generates new genetic combinations leading to variation? Yes, this we can understand. Vegetative propagation will not cause any genetic variations. Parthenogenesis also will not cause any genetic variations. And this nucellular polyembryony also will not cause any genetic recombinations. But sexual reproduction will cause these genetic recombinations. So that's why the answer is option 3. Okay, next one. A true breeding plant is very, very important. What is the true breeding plant? Homozygous condition is required. So suppose when we are taking the homozygous tall. So now this is capital T. So when it is undergoing self-pollination, now it will produce 100% the individuals with same phenotype and same genotype also. This is very important. Here these are all tall and also having the same genotype. Yes or no? So this is very, very important. So this is called pure or true breeding. Yes or no? So similarly small t. Now this is also when it is undergoing self-pollination. It will produce 100% same genotype and phenotype like parent. So that's why this is pure or true breeding. So that's why this is called pure or true breeding. So here out of these options, so if we go through this, one that is able to breed on its own is not the true breeding. Please remember here, breed on its own means it is undergoing self-pollination. That is good. So if we clearly see capital T small t can also undergo self-pollination, it can also breed on its own. It is not true breeding. Yes or no? So here, produced due to cross-pollination among unrelated plants. How unrelated plants will give homozygous condition? So here these may not give homozygous all the times, and that too they are unrelated plants. So this is also not correct. Near homozygous. So they are talking about the homozygous condition, and produces offspring of its own kind. So offspring are also like the parent, genotypically and phenotypically also. So that's why this is actually defining the true breeding condition. Next one, always homozygous recessive. What about homozygous dominant? That they are not mentioning. That is also possible, no? So that's why this option is also not correct. So out of all these 4, the option 3 is the correct one, that is actually defining what is true breeding plant is. Okay, next one. If a colorblind man marries a woman who is homozygous for normal color vision, the probability of their son being colorblind is, please remember here, man is colorblind but the woman is normal. So now we will see regarding this one. Genetic cross we have to do here so that we will understand clearly. Woman is normal. Please remember, color blindness, gene for color blindness is present on X chromosome. So that's why we are directly talking about X chromosomes. Is married to a man having the color blindness. Please remember here. So in the males, 1X and 1Y are present, no? So whatever is the X that is present, the same is having the gene for color blindness, and we are indicating this with XC. So now here what happens? Now X and XC and X and Y. XC, X and XY. So in this way, if you are writing X, XC, okay, and then X, Y, X, XC and XY. So what they are asking? They are asking about the son being colorblind. So here who are the sons? Female, male, female, male. So son and son. So are the sons colorblind? No, they are not colorblind. So please remember here, whatever is the X chromosome that is present in the sons, this is actually obtained from the mother. This is never obtained from the father. Y chromosome is obtained from the father, so X chromosome is obtained from the mother. So that's why if the father is colorblind and the mother is normal, sons can never be colorblind. Then what about the daughters? Daughters are also not colorblind. Please remember, some students are thinking that heterozygous condition is also colorblind. No, they are genotypically heterozygous. We are calling them as carriers, but phenotypically they are normal. So in this generation, no one is colorblind. All of them are normal phenotypically. Yes or no? So that's why what is the answer for this one? It is 0. Sons are not colorblind. Yes or no? So with this, we have successfully completed the revision of previous questions from this chapter. Okay. So in the next lecture, we will meet with another important chapter. So thank you so much, my dear friends. Thank you.